Trying to rock out here as we start up This Week in Science, our weekly podcast broadcast. It's me and Blair tonight. Justin has gotten a job with a new schedule that conflicts with the Wednesdays here of This Week in Science. So we'll see what happens with that as we move forward. But in the meantime, it's me and Blair. And you. Thank you for being here for another episode. As you know, this is the part that will be chopped out for the podcast, as will all the other bits of problems and stuff that occur. Hopefully none of them do, and that this is just a problem-free show, and we are fantastic. Indeed, right,
Consummate professionals. Yes, absolutely.
Yes. So pro. But in the meantime, the podcast is where the edited stuff goes. This is where you get all the stuff, warts and all, problems and more. And this is where we chat with you, and it's a really lovely experience for us all to be together. Make sure to hit the likes, hit the shares, get us in the algorithms, and let's be watching and talking together. Ready for the show?
Let's do it.
Okay, let us begin. And I'm scrolled all over the place while we were waiting. I had everything set up, and then I'm like, oh, scroll. Hello. Starting in 3, 2. This is TWIS. This Week in Science, episode number 1033, recorded on Wednesday, October 8th, 2025. Free to be science-y. Yeah. Hey everyone, I'm Dr. Kiki, and tonight we will fill your head with cheese feet, blind spots, and yogurt. But first, disclaimer. When universities bend the knee, what happens to the idea of being free? When science funding is based on politics, what happens to the lot of us? Academic rigor goes hand in hand with freedom. To self-police is required. But when thought police regulate previously free men, what will we talk about on This Week in Science? Coming up next.
I've got the kind of mind that can't get enough. I want to learn everything. I want to fill it all up with new discoveries that happen every day of the week. There's only one place to go to find the knowledge I seek. I want to know what's happening. What's happening? What's happening this week in science? What's happening? What's happening? What's happening this week in science?
Good science to you, Kiki.
And a good science to you too, Blair. And everyone out there. Welcome to another episode of This Week in Science. We are here to talk about all the science that we wanted to bring for the show this week. And unfortunately, we are missing a Justin, but I guess we won't have a This Justin. Oh, I don't know.
You'll have to catch us up later.
Yeah, we'll have to figure something out. We will make it all work. On this week's show, I have a lot of science news. A lot of things happened. We got some little awards, prizes that were handed out earlier this week. Some news stories about a new old yogurt recipe. It's a little spicy, a little exciting, a little dangerous maybe. And I also have Alzheimer's.
Oh, that's interesting. I also have Alzheimer's, but dolphins with Alzheimer's. We'll get into it. What a coincidence.
Yeah, this is Alzheimer's week at TWIS. Yay.
Yeah.
Blair, what else is in the animal corner?
Well, I also have a very fun story about leafcutter ants. And then I have 2 different spider stories for the kind of the quick news segment at the beginning there that I'm very excited to tell you all about right before you go to bed in many cases. Or while you're eating dinner. In both cases, not great. I can't wait. I can't wait to get into it.
I can't wait either. There's lots of spiders. It's spider season where I am. They're all around my door, around my house. My family has finally gotten to the point where they're like, Mom, Keeks, can we like clean up the spiders a little? And I said, no, you have to wait until after Halloween.
Yeah, this is when the spider females get really fat and pretty.
So pretty.
And the
Especially
most
the orb
terrifying,
weavers.
also, my personal least favorite, the orb weavers. This is when they're out. The females, just huge females, making very thin, very invisible webs right at face height.
In front of your
Yeah.
door for when you're leaving.
In front of
Yeah.
doors, under awnings. Truly awesome and terrifying.
Wonderful. Just know they don't want to end up in your hair. If you hit their strand that does cross your doorway, they're more likely to drift on the remains of the web to go back to the support column as opposed to you. They don't want to end up in
and
there.
a lot of orb
It's not-
weaver species also, my favorite thing about them, in an otherwise less than favorite bag of tricks, is that they will often remember if you disturb a web very tactfully in a particular place, they will not build it again in that same place. They know. So if
that's
you
why
get
fire.
if you get a face full of spider one time, they will not rebuild in the same place. In most cases, for orb weavers
to know.
specifically. Asterisk.
There are more out there. And there will be more science of spiders tonight. And if you want to share the spider science with your friends, let people know that they can subscribe to This Week in Science. Most places where podcasts are found. We're on all the big ones and a lot of the little ones too. Wherever you like to find podcasts, look for This Week in Science. We also stream weekly on YouTube, Twitch, and Facebook. And you can find information about the show at TWIS.org, twis.org. Now it's time for the science. You ready?
Yes.
Yay,
Let's get into it.
It's award season.
Yay! What did everyone
And-
wear?
I wasn't paying attention to that. I'm excited about who won for what. Oh my gosh. Nobel Prizes were handed out this week. It is the big week for the it's not the most money. There's other new prizes and globally and that are more splashy and funded by tech people and whatever. But this is an old, solid award. The Nobel Prizes, given to 3 people in a number of areas of science: physics, chemistry, and also physiology or medicine. This year, the Nobel Prize in Physics went to an area that is a little confusing. It's not really something that we can see in the world. It can't really be described by macro physics or, just doesn't quite, doesn't work in the same way. Quantum physics. Yes, these researchers, John Clark, Michael H. Deverett, and John M. Martinez, they all won for the discovery of macroscopic quantum mechanical tunneling and energy quantization in an electric circuit.
Okay.
Why? What does that mean and why? This is like the big question, right? It's what does this do for us? So one of the big questions about how energy in a circuit works and how we can measure voltage and how we can use the understanding of quantum dynamics and these quantum weird things like tunneling to be able to understand things at these levels of quantum computing, quantum circuits, at for things related to photonics. There's all sorts of levels where quantum physics comes into play. And so in what they did is they were able to make a device that like can fit in your hand, a small device using material that is superconductive in nature, which means that it allows current to flow without resistance. And this device that they created, they set up so you know how you like in school in the old days, they're like, let's make a simple circuit. And so you start like with a battery and you have like a lever that like turns it on and off that like opens and closes the circuit kind of thing. Like if you can imagine that kind of a circuit, but instead of like bigger, what they tried to do with instead of copper wire, using this semiconductive material as the conduit, what it does is when these atoms are, or when the current is turned on, it all turns into basically one electron. As opposed to when you think of current, it's like electron, 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 jumping, 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 running. You think of like separate ions running through a current as separate things. Because of the way that the quantum physics of this situation works, the ions, when you first put them in, they become these Cooper pairs, where the pairs become one. And so two ions act like one. And then when you like basically go, hey, turn on, the whole thing, it's like it all stretches and it's one ion for the whole thing. So there's no voltage. So even though you like were like, hey, kind of go, there's no voltage flow. Yeah, so it's this really weird situation where it's like you have all sorts of ions, but suddenly because they're in this like wee water slide, they're like, water slide train, and we're a worm. I don't know. I just made that up. It probably makes zero sense. Anyway,
it's like when you scrunch the outside of the disposable straw, and then you put a little bit of water on it. It goes.
Yeah. Kind of, but instead of like it's like extended, like you had a small thing.
No, that's what. Yes. So you scrunch it up. You scrunch it up on the straw, and then you take this and you go.
A long time.
Yeah, and you let a little bit go, and it goes, and it expands out. But this is more of a.
Yeah, and so the situation. And you know how we in the regular macro scale,
I love this graphic.
I know regular physics, you have like a lever. I'm closing the circuit, so current can flow. Opening the circuit, no flow. Well, they have a different kind of little lever in there. It was a junction called a Johnson gap, and that particular gap created a barrier to flow. But it was like this weird, it's a weird thing, and I don't understand it enough to explain it. But what it allowed is that they turned a voltage on. They were like, okay, we're going to see if there's a difference in voltage. We're going to see if current is changing, if there's a difference in voltage. Because right now, we have one ion going all the way around and then kind of stopping at the end. It's not flowing past this Johnson bridge. But because of quantum tunneling, which is like nuclear radiation, where like eventually these ions are like, I'm going to go through the barrier. And it's probabilistic, right? So there's math involved related to the probability of the density of a nucleus and the amount of current and, the barrier itself, how thick it is, how hard it's going to be for any kind of nuclear radiation to pass through. But tunneling is the process of that ion on one side going bloop and getting to the other side, even though there's a wall in the way. So it doesn't work in the real world. If you throw a ball at the wall will bounce back and hit you in the head, maybe. In this particular situation, you've got a whole bunch of pushing on the wall, and it's like, wait, oh, and one makes it through. And so eventually they were able, over multiple trials, they were repeatedly able to show that there's a consistent and deterministic kind of, it's like the math works out that you're like, oh, we have this, we have that, this is how it's going to work. And the math all works out in the way that's predictable for the current to flow. Okay. Why is this important? Because it's creating miniaturization. So this is a handheld device that they made, right? So this is reducing the size of quantum circuits and detection. This was creating an understandable. It was validating understanding of like these really crazy aspects of quantum physics, which when you get in the quantum scale, it doesn't make sense. It's not like our world. Yeah. You're holding your face. Why are you holding your face?
I think I kind of get it. I kind of don't. Like, where is the power coming from if it's just one electron? I don't know. That's why they got the Nobel Prize and I didn't.
Yeah. So the mechanics of quantum tunneling are it's not something I know well.
Yeah.
But being able to understand the practicalities, that the theory behind what they thought, they were able to actually show it working in a practical sense. They were able to use macroscopic tools to create a measurement system for this microscopic or quantum level phenomenon. And so this has lots of, it's already, this is not something that was just done. This is work that has taken place in previous years. And so what it's been able to do is that it's been able to underlie and undergird other types of quantum mechanical experimentation, development of technologies, understanding further these phenomena that are so very weird. That don't make sense in our world, right?
I just think about the notebooks and notebooks full of math that led to them thinking this was possible.
So many chalkboards, whiteboards, so many things.
Yeah. Just that we're seeing them say like, I think this might work. And then they get it to work. But the amount of work that gets to the, I think this might work is a mountain. It's like the iceberg thing, right? That's everything under the water that you're like, I have no idea. It's,
And
anyway.
to go from the math and the theory to an experiment, right? Say, I know we can test this now. And so that's what they all did. Different years, 1984, 1985. It was a series of experiments that were conducted at UC Berkeley. And this is the big thing that's happening. The Bears, go Bears, at Cal Berkeley have been putting Nobel Prize winner stickers on their football helmets for the football games this
Very
year.
good. So
great.
Forgive me because I forget we only do this once a year. Is this a normal timeline that a 1985 study is getting their Nobel Prize now?
Yeah.
Okay. Yeah. So that's part of the conversation that we have every year of how it's so white and it's so male, and it's because there's this huge lag.
Except for sometimes, like you get stuff where it's Doudna and Charpentier who, and I'm forgetting the Harvard, the Broad Institute researcher, and I'm David. I'm forgetting, but their research for CRISPR that was much more recent, right? So we also had the mRNA vaccine for COVID that was much more recent. So there's some things that come super, super fast, and then other things because they're so apparent, and others that don't. Moving on from physics. Oh, also one of the no, this is for this next one, the chemists. I do love the chemists. So the next award is for the Nobel Prize in Chemistry. No, did I miss one? Wait, no, he's the last one. Ha ha ha. Physiology and medicine. That makes sense. Okay, Nobel Prize in Chemistry. We have Susumu Kitagawa, Richard Robson, Omar Yagi. And these researchers received the Nobel Prize in Chemistry for the development of metal organic frameworks. And if you have been listening to the show, watching the show for a long period of time, you'll know that we have discussed metal organic frameworks and also other types of organic frameworks as in with respect to how incredible they could be, the potential for what they could do as tools, chemical tools for us. And one of the phrases that I thought that the Guardian used, I think it was the Guardian, basically said, oh, researchers won a prize for Hermione's handbag, which I think is fantastic because these metal organic frameworks, they set the basis for creating chemical architecture or frameworks, boop, 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 within which the hollows inside of the frameworks can trap other atoms, ions, things, and also allow chemistry to take place in a concealed place within. So they're very, very cool and have so many different applications, from environmental remediation to atmospheric to so much. They're even MOFs, metal organic frameworks. I think Justin had a story last year or the year before about how they were going to revolutionize biotech, which I think is also, and medicine, which I think is also very interesting. We're using MOFs to harvest water from desert air. They can capture carbon dioxide, store toxic gases, or like I said, make little rooms for chemistry to happen. And we've spoken with researchers in the past who have worked on offshoots of these, who are potentially creating other kinds of frameworks that could be used to clean our water, to get PFAS out of the water, to be able
That
to,
would be great.
so many amazing things could be possible. Right. And so this award this year, the researchers, Susumu Kitagawa was working on this area and came up with, I think it was Susumu who came up with the ideas first. Maybe I'm getting confused. I'm just very excited about the Physiology and Medicine Award that's next. But anyway, rooms for chemistry inside molecules. That is the basis of this. And so I actually think it's a really good award because MOFs have been a jumping off point and will continue to be for some very impactful science for a long time to come. And our one female science winner is within the physiology and medicine category, Mary E. Brunkow, and Fred Ramsdell and Shimon Sakaguchi. They have won this award this year for their discoveries concerning peripheral immune tolerance. Now, what does this mean? Peripheral immune tolerance. Why doesn't everybody have autoimmune disease? Basically. Like, why doesn't our immune system attack our body all the time? We've got these cells roving through the body going, you're an enemy, you're an enemy, you're an enemy. But what is it about? What are the ones that are like, ooh, you got that wrong. Nope, not an enemy. What are the ones that are like,
here. I go here.
Yeah, or the ones who are like regulating and saying, hey, no, you're too excited. That is not an enemy. It come to turn it off. It turns out that Shimon Sakaguchi was working on this area of T-cell research and was kind of off in left field doing some work that not a lot of people were looking at, but had these questions related to T-cells, and had an experiment where he found a certain type of cell, these regulator T cells, or now as they call them, TREGs. But he discovered this type of cell, and it was like, this is interesting. When I, take this out or put it back in, oh, suddenly things have changed. And then Mary Brunkow and Fred Ramsdell, they did some further work and were able to isolate the genes involved, the Circ genes that were involved in actually leading to the creation, and which individuals have them more often or don't. So like autoimmune, people with autoimmune disease tend to not have a great TREG system. And then Shimon Sakaguchi did another study and really nailed it down and was able to say, hey, these regulatory T cells are the thing from our thymus and are so important to making sure that our body doesn't make mistakes, and has led to an incredible field of work determining the development of treatments for autoimmune diseases, for cancer, for a number of immune related issues. So anyway, how is the immune system kept in check? It's the tregs, man. The tregs. T-Regs.
And so Zombie Tom Hanks in the chat room asks, can I CRISPR my allergies away?
Not yet. Not
No.
yet, unfortunately. No.
No.
Maybe with those tregs and the CRISPR, we can put these pieces
Put
together.
it all together. Yep.
Yeah, I like it. But anyway, these are the awards this year. And at first I was thinking, okay, maybe this year they played it kind of safe. It's not really any big outstanding science that you'd be like, yeah, I knew that's what it was going to be. But at the same time, all of the choices this year are incredibly impactful for providing steps in the stairs that we progress along, right? And the final aspect of this story that I absolutely love is that Fred Ramsdell, who helped with the tregs, he was on a digital detox in the Western US without his phone, and nobody could get in touch with him to tell him that he had won the Nobel Prize.
That's funny. He was just like camping in the middle of the wilderness.
Yep, he's off.
That's
He's doing, and like I love it. There are articles like scientist living his best life
also wins Nobel Prize.
Doesn't know he's won Nobel Prize. Nobelity. But he came back and he said that it was a wonderful, wonderful surprise. His wife let out a yell. This is a story from the Guardian. He feared she'd spotted a grizzly bear, but she's had like 200 messages on her phone saying he'd won the Nobel.
When was their study from? Do you know when that was from? I'm wondering how long this has been like sitting on the shelf and they just all of a sudden got this call.
This is again work. Okay, no, that's Metal Organic Frameworks. Hold on, moving backwards. Gonna get the right paper out here. So this work was completed. Sakaguchi was his work was 1995 initially.
Okay.
And so and then Brunko and Ramsdell had their next work in 2001, and then the final work that was it was a FOXP3 gene that was governing the development of these cells, and that was nailed down after that in 2003 by Sakaguchi.
So it's been over 20 years. That's kind of what I was curious about, because it's not like you like go out of town when they're announcing the Oscar nominations or something like that. You don't know. You're not going to schedule your whole life around the Nobel Prize announcements every year for infinity.
And if you've done the kind of work that people think is impactful, you've probably been nominated a couple times. Maybe you're like, I'll be nominated, whatever. Maybe you don't even know. Maybe you've been living your best life all summer.
Yeah. For real.
Maybe we should all digital detox a bit.
But not right now, because you're watching
not
the
right
internet.
now. Yeah, thanks for joining us for this show. So anyway, there's a lot of treatments that are coming out of this using these regulatory T cells that are undergoing clinical trials currently, which is pretty cool. All right, and some other interesting medical kind of physiology news, not an actual in the clinics yet, anything that doctors are going to be using, but there was a paper published in the Journal of Translational Medicine this week by a group of researchers who have validated and developed a blood-based test for basically chronic fatigue syndrome, or for people who are suffering from long COVID, MECFS, myalgic encephalomyelitis. And this is currently the only test that is barking up a tree to be approved. And this is the first publication that really makes it look like it's going to be good. It has a specificity, the probability the test will rule out negative cases, of 98%, which is very high. And it has a sensitivity, which is the likelihood of a test being positive that if that patient has the condition, of 92%. These are blood samples from still a limited group of individuals, but there are
Humans
questions.
was my question. Yes. So
they are
did
humans, people.
Okay, great.
No, they're
Yes.
This is past mice. This is into
Yes.
humans, and they have
Great.
they tested it on blood samples from people with ME/CFS, people that didn't have it. And the cool thing about this that I think is neat is this is looking for epigenetic telltales. So when we're talking about genetic stuff, like genetic stuff is you're born with it, right? You have that mutation. These are the proteins that are whatever. But epigenetics are those little tags and instructions that change and influence things during your life. And so they have found a suite of markers for these epigenetic things. They're using what they call epi-switch, epigenetic markers. These are unfixed. They can change during a person's life. And that's how they were able to get to such a high level of accuracy, sensitivity, and specificity. But this needs more testing. It needs more validation. This is promising because I think there are a lot of people in the world who would really like to be able to say, can you validate this? Like,
Yeah.
is this just a weird set of symptoms? Like, what can you like at least categorize this? Can you put me in this box, please? Because
Well,
it would explain a lot.
I can imagine a situation where a person is going to their doctor over and over and over, and they're being told, "Oh, you're probably low iron. Oh, it's because of your hormones. Oh, it's because you're not eating right. Oh, you need to get more sleep. Get a new mattress." Just like all these things, right? That happens when you go to the doctor and you complain about an issue that you've been dealing with for a long time that could be chronic, right? Being able to say, "Sure, I will do those things, but please get me the test." That's going to be able to be something that people can use to advocate for themselves, and that'll be a much steadier rate to, treatment than any of these other kind of Band-Aid suggestions.
Yeah, and we were talking about chronic fatigue syndrome when I was a graduate student back at UC Davis. And it did not have, people were coming, and people were considered quacks. They were fringe scientists. There were who are you trying to call this a syndrome?
Oh, she has the vapors.
Because it is very often women, not always, but immune systems for women are different because pregnancy and, being a woman and all that.
And all the hormone changes
and all the hormone changes through life. And so there's a delicate dance at play. But yeah, vapors, you're hysterical, whatever. It was for a very, very long time, chronic fatigue syndrome wasn't even categorized. And they did change it finally to ME/CFS, so the myeloid encephalopathy, I cannot remember, which I appreciate the acronyms for this. I'm sorry for those of you who may be struggling with it. But the ME/CFS and ME/CFS chronic fatigue syndrome has come together, and specifically since COVID and long COVID, and so many people having this syndrome and having to deal for a very long time with these symptoms. And we now have a media environment where people can talk about it and make it public, and suddenly people are paying attention. And I think it's a huge step. And so I'm like, get this, make this test work, make it go. I don't know. Blair, do you want to tell me about spiders?
Yeah, cheesy spider feet.
Yes.
Okay, stand by. My computer is making me reload the page and it's glitching, and I don't want to turn into a robot. Okay, here we go. So, cheesy spider feet. Black widows. Would you guess that female black widows smell like cheese feet?
Nope. I never smelled
Yeah.
black widow spider's feet.
So, a bunch of researchers looked at black widows, Latrodectus hesperus, and they are unique in many ways, but in this particular context, they're unique in that they use scent to lure in males to their webs. What they do after that, we've explored thoroughly on this show. Look it up. There's some black widow mating is some of the most wild things I've ever talked about on this show, but I won't kind of digress into that for the next 10 minutes. Instead, let's talk about how they smell like cheesy feet. So the females can lure males into their web via a chemical pheromone. What they do is they actually apply a chemical pheromone on their web, and it decomposes slowly over time. And as it decomposes, the scent becomes stronger and stronger, and that creates an attractant that can last for weeks, which is the main deciding factor in which female they're going to go to, is like, how smelly is this web? So a lot of other insects release pheromones from their body when it is time to mate and when it is time to breed. But these females, they use a long-term scent transmitter, so it lasts for all this time. So they're not just like, oh, there's a male, let me attract him over here, right? So they are able to do it and just kind of like apply the dressing and relax, and then the males will come to them. On direct contact, they trigger a charismatic mating behavior, so then they can also, like, when the male gets close enough, just kind of like whap, and then like, oh, you can't help it now, bud. But also the ability for them to decompose allows them to release this aromatic attractant, and the scientists have said that the attractant smell reminded the researchers of cheesy feet. Now, let's take a quick detour here.
Yep.
I want a word with these researchers. Out of the entire world of smells that you could pick from, you're choosing something called cheesy feet. Now, first of all, what is cheesy feet? Because I have an idea of what it might be, and it's really, really gross to me. And I don't even really want to describe it, but I'm picturing like somebody who needs to wash, right? And like,
Yeah, it's like the socks or the shoes, but it's like the smell kind of like a cheese rind that's gone too far, maybe? The moldiness.
yeah. So, but this is
also
what I'm saying. Like,
foot
they
funk.
could call it like, oh, it smells like an aged camembert, or like it smells like rotten eggs. Or it's cheesy feet. This is what you have to.
Does this come from wine tasting? Is my question. Like, is that
there
where...
cannot? I do not believe there is any way, anywhere in this universe, that anyone uses feet as a descriptor in a wine varietal. That cannot be true.
You don't know wine people.
I mean, I went to school in wine country. I'm pretty familiar. I don't recall feet being on the menu when we were talking about like hints of things. Anyway, okay, so I take issue with this particular terminology. I really don't understand why they picked that. This feels like this is like a deep-seated memory in somebody's brain from like childhood where their brother put socks on that had cheese in it, and that's what it smelled like. You know, like it feels like there's some backstory I'm missing here, because like cheesy feet. Okay, anyway, so
it was probably something
stinks,
from a sleepover prank gone wrong.
Yeah. So it stinks. That's what we're gonna say. To humans, stinks. Don't like the smell. But to male black widows, very attractive. Now, the other thing I wanted to mention here, and I'm gonna make some jumps in logic here, so bear with me. So in the research, they found that females adapt the intensity of their scent signals to the time of year. Now, normally, Other insects that release pheromones would release pheromones at the time of year where males are trying to find a mate and breed. But the female black widows are doing this year round. They're doing it most when it's mating season, but they're doing it year round. Kiki, do you think of why that might be? It is not in this article or in the paper, but I think I know why.
Why they would be looking for the stinky cheese feet?
No,
Year-round?
why they'd want to attract males at times other
To
than
eat
mating.
them.
Yeah, that's my assumption.
It's called nutrients. Yes.
Yeah.
Thank you. You weren't thinking in the right time of season, but thank you.
Yeah. I'll
Yes.
eat that.
The early spider gets eaten. That's all.
Yeah. So that's what I'm guessing here. I don't know that for a fact, but that's the leap I'm going to make based on what I know. In further research, they also were able to kind of figure out how they made this sticky cheese smell. And then they even were able to make a synthetic scent that attracts males as well in the wild. So they're narrowing it down. They're figuring out what's at play here. And that's, of course, important for invertebrate conservation. It's important for understanding mating strategies and movements of black widows. It's important for understanding how to manage black widows around humans because they're dangerous, right? So there's lots of different ways that you can kind of learn from an animal like a black widow and their mating habits and use that information to everyone's advantage. But stinky, cheesy feet webs. Let's hope Spider-Man's webs don't smell like that.
You never thought about it, right? Suddenly it's like,
I'd assume that spider webs don't smell. The idea that a spider web smells is truly wild to me, and now I will always wonder.
Wafting.
Yeah, like
Walk
Peter
the
Parker
web.
at least makes his webs like in the lab, and so he can choose to add a scent compound or not. I don't think there's a lot of accidental smell, but considering that like the Tobey Maguire version of Peter Parker creates them naturally, they can smell. They're just coming out of his body. They could totally smell.
Yeah.
So think
Wild.
about that.
So black widows can smell. They are attracted to certain smells. And if you do not want to have male black widows or female black widows, for that matter, around you, please avoid the cheesy feet odor in your house. So you might want to talk to your child.
Or your podiatrist if you're the problem.
Maybe look in
Who
your
knows?
cheese
Why
drawer.
blame your child? Maybe it's your feet.
Maybe it's your feet. Oh, okay. Speaking of wonderful things to smell, that leads to things to eat. And I have a couple of quick stories related to some really neat work that came out this week related to people and things we eat. There is a group of people, the Turkana people of Northwest Kenya, and they exist in an incredibly dry area that does not have a lot of vegetation. And so they rely predominantly on milk, meat, and blood from herds of camel and goat for their diet. Not a lot of veggies in there. 80% of their diet consists of animal products. And we know, here in Western world, people are like, yeah, eat animal products is good for you, whatever, keto diet, etc. But that causes actual heart problems. You can get gout. There are issues related to your kidney function and your metabolism that lead to various serious health issues if you're doing an 80% meat product or animal product diet. It can lead to some, over time, serious issues. So these researchers just published their work, and it's really wonderful because the researchers worked with the community, got their permission, their consent to work with them and do this a study because in modern day Kenya, people leave the Turkana area and can head out to cities. And if this is whatever, 80% animal products, whatever, if that's just a thing, whatever. But if it's going to cause them health problems because they've adapted over thousands of years to their environment and this diet, then it could help their people. So the researchers found that a majority of pastoralists were found to be chronically dehydrated, but very healthy. They looked at the genes with other indigenous communities in the region, 8 million gene variants or so, and they found differences in DNA differences, consistent DNA differences, excuse me, in 8 areas. And one is around a gene called STC1, which is related to how the kidneys retain more water. And so STC1, when the gene gets turned into a protein, it can lead to proteins that help to conserve water, and it also can lead to potentially protecting the kidneys from the waste products that come from the high intake of meat. Gout is not uncommon, or actually, no, I take that back. Gout is not common among the Turkana people. They are living dehydrated and meat happy, and they're fine. And basically, the evolutionary story is that these people have been, for the past 5 or 8,000 years, existing in these conditions with as much of this meat in their diet, and that it appears that they have adapted over time. And so this study and others like it may help keep the people in their in their youth, those who decide to go live in urban centers, from being sick, or when they go to the doctor, being able to say, this is wrong with me because this, or maybe being able to have natural diet interference that they are able to do. But anyway, they were able to show that this STC1 is changed in the Turkana versus other people, and it's pretty cool. I mean, you and I would have issues, but it's pretty neat.
Yeah, I mean, humans are animals. And animals in very specific extreme environments with very different habits and diets than populations of the same species that live elsewhere adapt to that diet. So, like,
but
whether it's something that's happened over many, many generations and
Yeah.
is related to gene expression, or whether it's something that has to do with how the baby is initially raised from the day that they're born, or if it has to do with the microbiome that they're exposed to from mother's milk, from their habitat, from, you know, the birth process. Like, those are all things that go into that in nature in any animal population. So it does kind of, it tracks. But it's, when we think about it in human terms, it sounds wild.
Yeah. Yeah. I mean, it's just hard to believe that we are isolated enough here and now that we can still have these kinds of differences.
Yeah.
Well, we do, and it's so neat. And it's helpful to know about it, especially as we meld and merge even more. And
well,
then,
I think about, like, even
Mhm.
real quick, like when you travel to a place and you get sick from the water, and the people who live there aren't getting sick from that water every day, and you adjust to it over time. This happened when I moved to Israel, 12 years ago. I got so ill for like a whole week, and then it was fine for the rest of the time I lived there.
Yep.
It's just something that, everyone else's biome has adjusted to, and that's a very, like, simple adjustment, but it's kind of a similar scale or reminder that there are variances, even though we're very, like, interconnected and interwoven, and there's not a lot of, like, super isolated communities. There still are differences enough in biomes that you can kind of see that. This is really cool to kind of see happen.
And I think like we go, oh, camels, they can exist where there's not a lot of water. You know, we're like, oh yeah, people, they just have tents and clothes that help, right? We don't think about the actual human adaptation because there is still this Western separation of us from animals, right? And so animals can be adapted to dry places,
Mm-hmm. Mm-hmm.
but we have to make our adaptations. Like that is the status quo way of thinking about humanity, right? We create our ability to exist, not, oh, we're an animal and we adapted too.
Mm-hmm.
In a certain area. So I think it's a very interesting juxtaposition. And then moving on from there, let's talk about yogurt in Turkey and Bulgaria. So the researchers publishing in iScience this last week, they went to the home of one of the researchers and investigated stories from ancestral stories that have passed down about recipes for making yogurt using ants.
What do you mean using ants?
Using
as an
ants
ingredient?
as an ingredient.
Nope. Must tell you, I am pro eating arthropods. Okay, I understand. I understand.
But all the times that we're like, oh, how did people figure out how to make yogurt? How did people figure out cheese? How did people figure out beer? All this stuff, right? Here's this method where the ants, with their formic acid, and they have a bacteria on them as well, which also helps to culture the milk from the goats and the sheep that they're using. And then the people actually take 4 ants from this particular kind of ant, and they put them in the milk, and they cover the milk with like a cheesecloth, and then put the milk with the cheesecloth with the dead ants into the ant pile, because the ant pile actually creates heat. And so the ant pile helps to culture and heat and culture without needing fire the creation of the yogurt. And so in a short period of time, this is a method of making yogurt using ants.
So not only are you using dead ants to seed the yogurt, but you're using the ant colony to heat it. Yeah. That's kind of nasty. It's kind of mean. Not nasty like gross, like nasty like rude. I killed some of you, and now I'm going to use your body heat to make food out of them. To
make the food. Yeah, it's amazing. So anyway, when I said formic acid, that is not the one that's right. It's the lactic acid, and bacteria create acetic acid. Let's see. So the live ants that they were using in Turkey and Bulgaria, like there are different species of ants that could be there that are around, and it doesn't really matter. But they were looking at these traditional practices and taking these historic practices that had been passed down in the Turkish mountain villages and other places in Bulgaria, and looking at how these yogurt making practices came to be. So they used red wood ants in this particular study, and they say that these countries are connected by cultural threads preceding the establishment of national boundaries, contributing to general continuity between their culinary practices. And so this is ethnographic evidence of ant yogurt across regions. And when did people go, ants in my milk, make yogurt? Yum.
Sure.
I'm
going to say, in the course of this story, I think you've convinced me. I'll try some ant yogurt. Why not?
I mean, seriously, it's wild. So they got a not popular, but a James Beard Award-winning kitchen to be involved in the research, and they were helping with the methodologies of what they were doing in these experiments. This they're like 2-star. It was a 2-star Michelin restaurant. I'll have to see if I can find the name of it
Wow.
again. But this group, anyway, they came together using science, culinary science, social science, looking at ethnographic and anthropological aspects of this to figure out what was going on. They did determine that it's the ants and the bacteria, so the ant and its microbes, that contribute to the acidification of the yogurt. And if they got rid of the bacteria or whatever, it didn't work the same way. Formica ants have the formic acid, so this is what was there, and this can be up to 10% of the ant's body weight, and so it can change the texture and the way that
It's more like a Greek style yogurt or something. Yeah. Okay.
And so formic acid might be doing different work. But the whole situation, they also said that don't do this at home. This has not been okayed as much as they've tried to okay it in Europe, because the ants that are used very often contain a parasite, which can cause health problems in people. There are ways to smush the parasite out of the ant and filter the parasite out if you are still wanting to use the ant and its bacteria and not the parasite to make your yogurt. Thank you, Frumpy Bee, alchemist in Copenhagen. That is exactly it. Thank you. Yes, alchemist in Copenhagen. And I honestly, when is Justin going to go back there? Because we should all go have dinner there and talk to them about ant yogurt, because I bet they have it.
Letter E here. That does not look like anything I want to consume. That looks really, really gross.
Letter E. Yes. So on the screen is an image of ants in milk covering.
Yeah, so that's
A. B is covering the cloth-covered vessel with dead ants with the ant colony materials. C is, I think, straining out the yogurt the next day. D is like, I don't know, some kind of a picture of an ant that doesn't make sense. And I could probably tell you what the captions say. All right. D, culinary applications created by the research and development team of Alchemist using Rufa ants. D, oh, here it is, ant yogurt ice cream sandwich. The top view shows the twill cookies that sandwiches the ice cream contained below. E is ant mascarpone-like cheese, and so that's not yogurt. They've let it go to a more cheese-like state. And F is a milk wash cocktail.
Nope. Nope. See, I wasn't okay with it. I was okay with it. I'm not okay with it again.
I think this is so great. But okay. It doesn't mean that we need to use ants to make our yogurt. But what I find fascinating is that this, as long as we have potentially had agriculture or had goats, sheep, cows, animals that have milk with them, at some point we figured out how to make that milk more conservable, right? And yogurt is one of those forms as well. It's not just for drinking. How do you eat it in different forms? How fascinating that maybe this ant process is something that someone noticed and played with. Ants in my milk. Oh, we'll see what happens. I think, the human curiosity and the way that we develop things that now we take for granted. Where did they come from? We didn't always have just, our yeast cultures for the sourdough. We didn't always have, the bacteria that were exactly right for whatever fermentation. We didn't have the bacteria for your yogurt. I think the process of all this and having this come from, his stories from, cultural stories, I think is fascinating. Yeah.
Hmm.
Apparently it tastes good. Tastes yogurty.
Okay. Noted.
Ants in my yogurt. I won't complain again. If there's ants in my yogurt, I might be happy. What do you want to talk about? Do you want to talk about your intersex spider?
I could, or we could just move into the animal corner. I don't know. What do you want to do?
Yeah, let's do that. Oh, and as
Okay.
a sideline, I'm just going to say, hey, we're connected by more than the internet.
What?
You and me, I'm in Oregon. You're in California in the Bay Area. I'm worried about the Cascadia fault. You're like, oh, San Andreas, that's a thing. Well, apparently a new Oregon study came out and said they might talk to each other. And so it might be like
Oh.
their little ruptures and disagreements. They're not just alone, that and when one goes, the other might too. So, hey,
Okay. Bolts be chatting. I got it.
yeah, pay attention,
Huh.
everybody.
Okay.
New study out of Oregon State University. Could be a bigger catastrophic thing.
We're all just floating around on fluid. Like, if one thing moves, something else has to move.
And we have to live in the pretty places. Let's live in the pretty places where there's food and it's dynamic. Oh, yeah, that's where there's volcanoes and earthquakes. Oh, no. Hey, everyone. If you're enjoying the show, please head over to twist.org, click on the Patreon link, and help support the show. I'd love to be able to say your name at the end. All people who support at $10 or more a month, I'm going to try and pronounce your name at the end of the show. I would love to include your name among them. It would be amazing to add more people to that list because I don't have enough of a tongue twister as it is. I appreciate everyone, no matter what, who helps us out, but it's fun to try and read your names and give people some credit. So if you can head over there to try and support us using Patreon, that would be fantastic. Also, if you need a t-shirt or a sweatshirt or whatever, head over to twist.org, click on that Zazzle link, and buy one of the awesome bits and bobs that we've got there. That'll help out too. All righty. Tell people to subscribe to TWIS. We can't do this without you. Thank you for joining us. We can't do it without you. And now it is time for that part of the show that we love to call... What is it called? Blair's Animal Corner.
She loves our creatures, great and small. Biped, millipede, no pet at all. If you want to hear about animals, she's your girl. Except for giant pandas and squirrels. There's Animal Corner.
What you got, Blair?
I have intersex spiders.
What does that
Yes.
mean?
Yes. So
What?
when I'm doing my research for stories, I see titles like that, headlines like that, and I go like, okay, so you found a hermaphrodite animal. Sure. There's lots of those. There's also like sequential hermaphrodites. I could do a whole show just about that and how normal it is and how we like stigmatize it as humans, but it is super duper normal to find intersex animals. And so like whatever. But I clicked on it. I was like, let's just see what this is about. I learned about a brand new type of intersex animal today. And I'm going to talk to you about it because I didn't know it existed. You might, Kiki, because you worked with birds. And it happens a lot with birds. But it is called a gynandromorph.
Yeah, I do know that one.
Yeah,
Yeah.
I thought you might because it does
I
happen
did.
in birds a lot.
Yeah.
Okay, so let me explain. So this all happened because in Western Thailand, scientists discovered a new spider. This spider was then presented to researchers. They were studied extensively. Many specimens were collected. And after analyzing the specimens under a stereo microscope and comparing them to similar species, just based on morphology, they have not done genetic research yet, but just based on morphology, they think they're part of the genus Demarcus, a group of Mygalomorph, which is a wishbone spider found in South and Southeast Asia. And so they're known for making wishbone-shaped burrows that they make in the ground where they wait for prey. And so they found that they have big differences between male and female spiders. The males are about 0.6 inches in length. The females are around 1 inch. The males are gray, and they're covered in a white substance that is yet to be identified. So you can think about that however you want.
Cheese feet.
Leave it to your imagination. Yeah. And then the females are brighter orange, and they have a white layer of an unknown substance. So what's crazy is they found amongst this collection a gynandromorph. A gynandromorph is an organism that is half male, half female, as opposed to a hermaphrodite where they have both types of sex organs. Gynandromorphs have they do not have bilateral symmetry. So hermaphrodites still have bilateral symmetry. They look like just anybody else, but they either present usually as female or male or a little bit of both, but it's like a mixture, and they are bilaterally symmetric. Gynandromorphs, one half of their body is male and one half of their body is female. Straight down the middle. And so this happens in birds a lot. This happens in crustaceans a fair amount, but I had no idea about this. And what's very funny is they actually named this new spider, the species Inazuma, which is a character from One Piece, which I do not consume. I'm not super familiar, but apparently it's a character that can change between male and female. And so because of that, they gave this spider this name. But I just wanted to kind of bring that up as a little sprinkle because a lot of people don't know about this very cool kind of sexual dimorphism. Or, sorry, not sexual dimorphism. Intersexuality. Something totally strange and unusual. Oh, Kiki, you're muted.
How does it end up hemispheric? Like, organisms like insects, arachnids, humans, we are bilaterally symmetric, right? And so there's the process of being like, this gene, we're going to mark that one not. And so there's a lot of picking and choosing and marking which of the fathers or the mothers genes get included on whatever side. But this is like, hey, you're half this, half that. There you go. It's amazing.
Yeah, so they have some ideas.
Yeah.
It's uncertain exactly how it happens. There's certain kind of theories in the scientific community that it might come from a loss of sexual chromosomes. So it might be an animal that is otherwise genetically female. It's a female zygote, but there could be like a nematode infection or some sort of microbial infection that causes a degradation of the chromosomes to make their body not develop correctly. But I don't know. It's very, very, it's unlike anything I've ever seen. And you can Google this. You can look at gynandromorphism, and particularly in birds, it's wild because you can see a bird that looks like a hen on one half and a rooster on the other half.
On the other, yeah.
Or like a cardinal that's red on one side and brown on the other. It happens in butterflies a lot. And I
just have to wonder, weird. Like, why say that it's a parasite or something else that, like, why not just one of those odd probabilistic things that happens?
Yeah.
Right? Like, why not just be like, it is? And
absolutely.
It's just, that's how they got marked. But yeah, I find it very interesting.
Yeah. Yeah, so there you go. That's your word of the week. Gynandromorphism. Next, I wanted to talk to you about leafcutter ants. And here's another, this is like the theme tonight, is that headlines. Let's talk about headlines. Here's the headline that I read. I almost passed it by because I was like, duh. Leafcutter ants have blind spots, just like truck drivers when they're carrying leaves. Duh. They're carrying a big leaf. Now, I read the story. This is a little more complicated, so let me get into it. So, leafcutter ants.
About the ants, their eyes,
Yes.
the way
Yes.
they're located, and
Yes. Yes.
all of it.
So
Yeah.
obviously they can't see around the leaf, right? So leafcutter ants, they carry a load several times their body weight. And what this research from the Smithsonian Tropical Research Institute in Panama was looking at is the quote unquote truck driver effect, which is when leafcutter ants carry an oversized load, which in some cases can be 8 times their body weight. They walk more slowly. The ants behind them, because they walk in a line, they're social, right? That makes them walk slower. And so this is just like if you're stuck behind a big rig on a 1-lane road. This is called the truck driver effect, and it can reduce the walking speed of following ants by up to half, by up to 50%. The reason behind this phenomenon is a mystery. Is it really just because they're walking slow because it's heavy, or is there something else going on? And so they recorded the ants with and without the leaves to see how they use the antennae. So this is it's not their eyes, it's their antennae. The antennae are in charge of touching, smelling, and tasting, and ants use chemical pathways to know where they're going. So they were measuring the ants and the leaves they were carrying and monitoring their antennae movement. And so then, based on their observations, they offered artificial leaves to the ants, which were tiny pieces of paper dipped in orange juice that made the paper attractive to ants. And then once they collected the leaf, they cut it with scissors while the ant was still walking.
Wait,
so they chopped part of it off while they were carrying, to reduce the load in about half. And then they watched the use of their antennae before and after they cut it in half. So what they found was that leaves did fewer antenna taps per step than ants without leaves. And that when they were cut in half, the ants increased their antenna taps. So basically because they were carrying something really heavy or awkward or whatever it is about it, they weren't checking where they were going as often. So this is the quote unquote blind spot that we're talking about here. It's not a literal blind spot. They could still see with their eyes, but their vision is very poor. They use their antenna to feel their way through chemical signals, and their ability to sense those chemical signals was reduced based on their payload. And so this means that carrying larger loads leads to difficulty with dealing with trail obstacles, surface irregularities, and it slows the whole colony down behind them. And this is actually exacerbated in larger ants, just like in a Big Mac truck versus a car. So aside from this just being interesting, it also does explain some phenomena that has been observed with leafcutter ants in the past, namely that they will sometimes carry a smaller load than they are capable. So people who observe these ants have noticed, like, oh, there's a bigger leaf over there. Why didn't you get that? You could handle this smaller leaf. Or you could handle the bigger leaf. It's not too big for you, but you picked the smaller leaf instead. And this might be why, is that they're choosing to pick a smaller leaf so that they can see where they're going better, move faster, and not slow their buddies down behind them.
Are you hearing the audio from the video?
No.
Good. Sorry about that.
Yeah, look at those little guys just rushing.
I remember watching leafcutter ants at California Academy of Sciences, and they're going down their path, and they've just chomped the leaves, and they are just doing their business, and they don't care about anything that's there. And so this explains that a lot. Like, they're just like, I'm carrying a heavy thing, I'm following my path, out of my way.
Yeah, we had, when I worked at the zoo, the leafcutter ant exhibit, there was one exhibit where the ants lived, and then they would drop all of the leaves and things in a different exhibit, and there was a tube that connected the two. So they had to travel to the second exhibit to pick up the leaves and then travel back. So you could get really up close to this tube to see the leafcutter ants up close while they were moving with the leaves. It was very neat.
That's so cool.
Yeah.
That's super cool. Yeah, I think leafcutter ants and these, we see these animals doing these things that are very stereotyped and just predictable. But then to find out that, what they're doing is actually something that is, it is really beyond what they should be doing.
Yeah. Yeah.
And it's
So wild.
overtaxing them. So it's like, hey, everybody, in your workday, when you're being overtaxed, think of yourself as a leafcutter ant. Realize you have blind spots.
Yeah. Yes, absolutely.
Your load.
Yes. And speaking of, I don't know. I don't have a good transition for this. Hey,
Oh.
marine mammals get beached sometimes, huh?
Yeah.
And we don't know why. It is kind of a phenomenon in the, biology community. Like there's lots of theories about why it might be happening, but now there's a new theory about why it happens in dolphins. Scientists in many different states, all the way from Florida to Wyoming, have worked together to come up with an unusual hypothesis, which is that just as some adult humans with dementia are found wandering far from their homes, dolphins might become disoriented because they are suffering from a form of Alzheimer's, and that is why they become beached. So this is... I could get very far in the weeds on this, but the long and short of it is they looked at the bacteria, the cyanobacterial toxins in the water column where beachings are more likely to happen. They found that those toxins are associated with misfolded tau proteins. That's the business of Alzheimer's. And also amyloid plaques. Also the business of Alzheimer's. And then they looked at kind of the neuropathology, the disorientation, the kind of the different, behavioral cues they were seeing, and it followed kind of similar expectations for Alzheimer's. They looked at this toxin a little closer. They looked at this particular toxin called beta N-methylanamo BMAA.
Favorite. Yeah.
Yeah. Which and also their isomers. And they found that they were very toxic specifically to neurons, and that when neurons are exposed to that, it triggered Alzheimer's-like neuropathology and cognitive loss in animals. And so also these toxins can be biomagnified. And what do dolphins eat? Fish. And so if there's cyanobacteria and the toxins in the water column and fish are eating them, then the dolphins are eating those fish. It comes extremely bioaccumulated in the dolphins. And so it would make sense that the amount of toxins that they have would just be off the charts. They looked at 20 common bottlenose dolphins stranded in the Indian River Lagoon in eastern Florida, and they found BMAA in its isomers. And they found that dolphins stranded during the cyanobacterial bloom had 2,900 times the concentration of one of the isomers than those from non-bloom seasons. So this is definitely bioaccumulating. It's happening in stranded individuals. And they also found brain neuropathology similar to Alzheimer's in those dolphin brains. So this looks highly probable. So now, of course, I want to know, let's go back. Let's look at all of these stranding events and beaching events where animals were doing it at just crazy frequencies. Was there an algal bloom at the same time? Do we have good enough data looking back where this has happened over and over?
Making it related to algal blooms, yes, that would increase the toxicity and all this stuff, but that it would be chronic, and exposure, and Alzheimer's, and beaching. It's just one time and done. Or is this because they've been through a number of blooms and it just, okay, this is the one that put them over the edge.
Right, exactly, yeah. Which, speaking of that, this is where it goes a little far afield. There's a lot of hypothesizing happening here, but dolphins are considered environmental sentinels or indicator species because of this biomagnification issue. And so specifically for toxic exposure in marine environments, when dolphins start dying, you go uh-oh, what's happening here? And so there are actually concerns that are growing about human health issues related to cyanobacterial blooms. And in 2024, Miami-Dade County had the highest prevalence of Alzheimer's disease in the United States.
It's not just people retiring there.
It could be. It could be reporting. It could be that people have better access to the healthcare that diagnoses that. It could be that the particular doctors in Miami-Dade County are more likely to provide an Alzheimer's diagnosis instead of other things. Like there's lots of variables. So this is where it gets a little less sciencey and a little more like, I wonder. But it is interesting.
You start going in on the variables and you can figure it out eventually. But yeah.
It is interesting because this is one of the areas that the cyanobacterial bloom was really wreaking havoc. And so it's possible if these people were going swimming in the ocean, Florida's beautiful beaches, they were not paying attention to algal blooms repeatedly over a long time, that they could be getting exposed to these things at a similar, not a similar level to dolphins, but still at a higher level than our bodies are used to, right? And so I think that this is not, oh my God, don't go in the water in Florida, you're going to get Alzheimer's. That's not the point of this story.
Not the story. No.
But I think there's a few points. One, we found similar kind of tropes to Alzheimer's in animals, which means we can follow research pathways with animals who exhibit Alzheimer's symptoms and neuropathology. Okay, so that's really important. We found a potential pathway to getting Alzheimer's. It is not the only one. Even if this is a cause of Alzheimer's, that does not mean it is the cause of Alzheimer's. Alzheimer's is kind of like a difficult egg to crack, I think, medically. We're having a lot of trouble kind of narrowing it down. But if it is a pathway, that is still beneficial to know about. And then also the piece about beachings and strandings, which I think is, a whole separate piece of these will happen, these events will happen. It'll be like, what is going on? What is happening to all these animals? So it's not that we have potential treatments for Alzheimer's in dolphins, but if you can kind of identify that there's an algal bloom happening, you can, I don't know, maybe if you have an endangered pod, you could try to move them, you could try to control the bacteria, you could try to recognize if an animal gets beached and this is what's going on with them. Maybe they need to be put in captivity instead of tried to be released into the wild, because it's going to keep happening, right? So there's lots of things that you can do with this information once you have it. So I think it's a pretty cool study with a lot of potential for more.
I think it's fascinating. And it ties into several of my studies. I have a couple on Alzheimer's actually. But what I find very interesting is the, connection to, it's not necessarily algal blooms, but toxins, right? So something in the environment that has led to a buildup or changed the way that the blood-brain barrier can deal with toxins, so that there's like stuff mucking up in the brain, and then the brain doesn't work right anymore. You forget things. The neurons don't work. There was a study this last week about looking at Parkinson's disease and particular proteins that are involved. Researchers actually caught these proteins I'm not going to say on camera, but they caught them in the process of puncturing holes in neurons. And every time they punched a hole, it was like neurons are like, spurt, I'm losing myself. But then the membrane would seal up. But then, if there's more and more of those proteins, they damage the neuron so much that eventually it just dies. And so if you have a situation that leads to a toxic buildup, right, where the body itself cannot manage the level of toxins anymore, suddenly the things are going to go downhill. The body is not going to be able to regulate that homeostasis to a point where it can get rid of the toxins at the rate that's fast enough or required. But all right, so do you want to say more about these dolphins?
No, give me the human Alzheimer's stuff. Let's go.
Okay. So you in this, you were talking about Alzheimer's. University of New Mexico Health and Sciences Center. Researchers just published a study in the American Journal of Pathology related to the blood vessels in the brain and dementia. Vascular dementia is a cause of dementia, and it's a problem related to the small blood vessels in the brain and how they get blood to different parts of the brain. I was looking at this particular study related to the dolphins, and I was like, huh, how fascinating. Maybe there's something happening to the blood vessels, or so where would the toxins be working, and how would they be actually creating this? But the researchers for this particular study, they found for humans and vascular pathologies, they hadn't ever really comprehensively defined the pathologies they were looking at that lead to vascular dementia. And they found that nanoplastics actually are very involved as a new player in brain pathology. Not just dementia, but Alzheimer's disease as well. She found that there are a lot more plastics. She says, quote, "What I'm finding is there's a lot more plastics in demented people than in normal subjects. It seems to correlate with the degree and type of dementia. The quantity of plastics was associated with higher levels of inflammation in the brains that she looked at." And this is the really first time that anybody has done a comprehensive look at neuropathology of the vasculature in dementias and Alzheimer's. And the bottom line is that the small blood vessels seem to have been punctured in the damage that is coming. So blood vessels have been impacted so much by nanoplastics that they have created inflammation so great that it's
Your
potentially
brain is just pierced by microplastics.
Yeah, and our brains, and if you imagine a dolphin or somebody who swims their whole life or whatever, the water is full of microplastics,
Yeah.
nanoplastics. Dolphins they're breathing, swallowing everything, right? So the nanoplastics, I'm thinking in addition to the algal bloom story that maybe there's more there. Just
Yeah.
if there is a general increase in this that's not related to the chronic aspect of the algal blooms, but I think these are bigger stories. And then the other story that I found is that researchers, and this is in mice, but this is the good news and hopefully something that can be very useful. Researchers at the Institute for Bioengineering of Catalonia, IBEC, and West China Hospital Sichuan University, working with other partners in the UK, have developed a nanoparticle therapeutic for Alzheimer's that targets the blood-brain barrier. So one of the things that has been found, and as you were talking about, it sounds like one of the things that has been found is that the brain and Alzheimer's disease with those amyloid beta sheets and all the tangles that come together, those amyloid tangles, that they occur at this interface of the blood-brain barrier where blood vessels have a mechanism that's usually guarded by a particular molecule, that LRP1, that grabs onto amyloid beta, binds to it, takes it across the barrier and back to the bloodstream so that it can go away. When amyloid beta is building up, it's potential that the LRP1 isn't working well. And so what they did is they created a mimic for the ligands of LRP1, and they injected it into the mice, and all the mice that they had that were a model of Alzheimer's disease, they have a genetic predisposition to have the buildup of amyloid beta. When they injected these nanoparticles into the blood of the mice got better.
Okay.
They did 3 injections over time. The mice started acting like not Alzheimer's mice. They started acting like wild type mice, which it's not. I mean, that's for their age, right? So it took it all back to the point. But anyway, this is in mice, and it's nanoparticles, but they did find that the nanoparticles are in the mice. They work well, and then they don't cause problems in the mice afterwards. We'll see if they can get this through the various trials that need to take place to see if they'll work for other animals and then humans. But it is a proof of concept for a new idea, which is not targeting those amyloid betas or the tau tangles or these parts that are kind of mucking it up to begin with, but targeting the mechanisms that help to clear the pathways.
Mhm.
And so, who knows? Maybe
Nice.
we'll see more of these nanoparticles in the future.
Yeah, and it doesn't have to be or, it can be and. So
Exactly.
yeah.
And
Mhm.
I mean, we've got some treatments-ish, right, that help slow down Alzheimer's a little, but like, let's get another, and let's help people because people are just getting older in the West. That's it. We're like swimming in plastic, so we're all gonna be demented, which would be really fun, possibly. Maybe that would be exciting. Oh my God. Anyway, last bit of happy joy news. Researchers at the Allen Institute have developed what I am liking to call ChatGPT mouse brain. In their
Okay.
story and headline, they decided to call it a ChatGPT-like AI model for neuroscience to build up a detailed mouse brain map. What they did is they used what is called a transformer model, similar to the learning model that's used for ChatGPT, which uses context. And so by throwing a whole bunch of brain cell data and into the mix, context was taken into account, and researchers can basically say, I want to know about this and what it's attached to and whatever, and it'll show you, oh, it's the hippocampus or whatever. And everything so far seems to match fairly well with historical expert-based brain nuclei that have been determined, but now it's got more detail. And so it's kind of it's a very exciting way to use spatial transcriptomics to be able to discover more about the brain. Only in mice. It's great. Maybe someday in people brains are a lot bigger, but in mice, it's great. Congratulations, all the people working with the Allen Institute. Yeah, it's huge. This is published in Nature Communications. It's 1,300 regions and subregions. This is UCSF also involved in this. Yeah, it's very specific. It's awesome. But that's all I have to talk about. So I've got some good news. You don't have any more scary spider nightmare juice, do you?
No, that's all I have.
Okay. Well, okay then. We don't
Which is
have.
good because I think I'm about to be summoned. So we should.
Yeah, it's time for us to make this move on. The summoning of the child has begun.
Yes.
Yes, we all have places to go, but what a wonderful, wonderful opportunity to spend a couple hours enjoying
Yeah.
science. Thank you so much, Blair. Thank you.
My pleasure.
Yeah. Thank you, everyone in the chat rooms. Thank you for being here and chatting. I know I didn't pop up your comments this evening too much on the screen, but I saw them. I see you. Thank you for being here and commenting and being a part of the conversation and helping us get through this show. I hope you enjoyed it. Fada, thank you so much for helping with show notes and social media. Such a huge help every single week. I know how much it takes. Identity 4, thank you for recording the show. Cord, Lore, others, actually everybody in the chat room, thank you for for making the chat rooms good places to be. We can't do it without you. And Rachel, thank you for editing. There are a few places in the episode tonight I hope you chop out. Not too many. It's all good. But we really appreciate what you do every week. Thank you so much. And as always, thank you to our Patreon sponsors. Thank you to Aaron Anathema, Adam Mishkan, Alan Viola, Ali Coffin, Andrew Swanson, Arthur Kepler, Artyom Brigg Call, Bob Calder, Bob Cowles, Brendan Minish, Brian Carrington, Chris Wozniak, Christopher Dreyer, Christopher Rappin, Craig Landon, Craig Potts, Dana Lewis, Daryl Myshak, Dave Wilkinson, David E. Youngblood, Don Mundis, Jonathan Styles, aka Don Stylo, E.O., Eden Mundell, Eric Knapp, Flying Out, Fred S104, G. Burton Lattimore, George Chorus, Greg Briggs, Jack, Jason Olds, John Atwood, John McKee, John Ratnaswamy, Johnny Gridley, Ken Hayes, Kent Northcote, Kevin Parrott, Chan, Kevin Reardon, Kurt Larson, Lon Makes, Lauren Gifford, Mary Gertz, Marjorie, Mark Flo, Noodles, Patrick Pecoraro, Paul D. Disney, Paul Ronovich, Philip Shane, Pierre Velazar, P.I.G., Richard Badge, Roderick Norland, Robert Farley, Rodney Lewis, Rudy Garcia, Remy Day, Sean LeCurrent, Slam, Shoe Bruce, Stephen Alberon, Steve Leisman, a.k.a. Zima, Sue Doster, Teresa Smith, Tony Steele, Vegard Chefstad. Thank you for all of your support on Patreon. If anyone else out there would like to have me trip over their name, please head over to twis.org, click on the Patreon link, and support us at $10 or more a month. That would really, really be a helpful thing. On next week's show,
we'll be back at 8 p.m. Pacific time on Wednesday, broadcasting live from our Twitch, YouTube, and Facebook channels.
Yes, and I'm reading further. Want to listen? No, Blair. Wait, yes, want to listen to us as a podcast? Just search for This Week in Science wherever your podcasts are found. If you enjoyed the show, get your friends to subscribe too.
For more information on anything you heard here today, show notes and links to our stories will be available on our website. That's at www.twis.org. You can also sign up for our newsletter that someday will come. It's like waiting for Godot. Yeah,
apparently
waiting for Blair's TWIS newsletter. You know?
We love your feedback. If there's a topic you'd like us to cover or address, suggestion for an interview, please let us know. You can tell us on one of our social media accounts if we're out there. Send us an email. Make sure you put TWIS in that subject line so that your email doesn't get spam filtered into a cheese foot spider web. Stinky cheese feet.
We look forward to discussing science with you again next week. And if you learned anything from the show, remember,
it's all in your head.
This Week in Science. This Week in Science. This Week in Science. This Week in Science, it's the end of the world. So I'm setting up shop, got my banner unfurled. It says the scientist is in, I'm gonna sell my advice. Show them how to stop the robots with a simple device. I'll reverse global warming with a wave of my hand. And all it'll cost you is a couple of grand. 'Cause this week's science is coming your way. So everybody listen to what I say. I use the scientific method for all that it's worth. And I'll broadcast my opinion all over the earth. 'Cause it's This Week in Science. This Week in Science. This Week in Science. 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 This Week in Science. This Week in Science. This Week in Science.
Oh, I had a whole song and everything. You missed it. Yep, muted. Sorry, zombie Tom Hanks. I saw the muted and I didn't even think that related to me. Oh boy. Here we go. Yeah, so I said this is the TWIS Book Club reading material. And then I was scrolling through the pages and I found out that I could take the mini course. There's a mini course that I could take. I love it. I will learn to be different. I will have a polarizing cult message. It's all branding. Anyway. Fun stuff. I'm interested in how things work in the world, you know. You want to bring the flavor aid? Sounds good. Yep. It's got electrolytes. Thank you, everyone, for joining us for another episode of This Week in Science. I don't actually know what's going to happen next Wednesday night. I said that we'd be back next Wednesday. But we have an issue in that Justin has a new job, and I am actually going to a summit, to a work trip. And so I am excited to be going there, but it's going to take me there through Thursday. And so we will either have Blair and somebody do next week, or we will call it off, or we will reschedule to a time when Justin can be joining us. Yes, you got what I was saying, Paul Disney. Yes. A couple of weeks ago, Justin came and said that he was applying for jobs, and I knew that the job he got could potentially change our scheduling. And so that is what we are going to be dealing with now. And Blair's also very tired. I don't know. Those of you who are here and watch every week, is Wednesday at this time the best time? Would other times be better? Would it be worth it? I know during the daytime, it's work time. Yeah, I don't know. It's an interesting question. I don't know what to do about it. So maybe each of us will have our own half hour alone with you during the week, whenever we can make it. Yeah. I don't know. I do love doing this, but I'm going to go. I appreciate you being here. And I guess there's nobody here to say goodnight, Kiki. Not Tuesday and not Thursday. Yeah. Okay, good. Yeah. Yeah. I don't know what days could work. Yeah, Justin his work schedule is a bit much. But that's what happens when you have kids in university. Oh, geez. That's a whole thing. Yeah. Justin, it looks like, is like Friday, Saturday, Sunday. Yeah, Friday, Saturday, Sunday. It's a problem. Stay. What am I gonna do? No, I have to go read my new material, because if Justin can't come back and I can't get a regular thing going on a week, whatever, if I can't make it go, well, I'm looking at job opportunities. Oh, flying out. I'm sorry, you're moving. Yeah, moving is always chaotic. Thank you for your flexibility. Fada, have a wonderful night. I need to catch up. I know Slow Horses is out. I love that series. Got to check it out. Have a wonderful night. Friday night. Yeah, exactly. We could go late and not regret it the next morning. Exactly. Yeah, that could be fun. I don't know. Justin had said Sunday nights, but I don't think I want to do Sunday nights because then it's just starting my week earlier, and then I have to put my son. He's 14 now and rolling his eyes at me way too much. So, oh boy. I appreciate you all so much. You have no idea. No idea. But let's move on. And if you want to email me or message me, however, let me know what you think. We're trying to figure it out. For the time being, I think it might just be I'm not doing anything, not making any drastic changes. It just might be me and Blair on Wednesdays. Maybe next week might not happen. I don't know. You're not opposed to Sunday either. Right. Yes. I have this very odd, like, since graduate school when it was like, I work all the time and everything is fine. And I spent a lot of time just like always working. And so I have this like boundary thing where mentally, psychologically, I'm like, let's make certain days not work days. And the weekends are that because that's when Kai's around. But yeah. Aw, thank you, Paul. I will. Yes. Advance notice. That's the thing. That's what I'm trying to do right now. I'm trying to be transparent and discuss just what's going on. And I don't know exactly what's going on. Justin got a new job, so we want him back, right? Right? I do. I'd like him back. So in order to make that happen, we need to make some little adjustments. So my Wi-Fi, it's so weird because I'm not on Wi-Fi. My internet has been so up and down today. I think we use NordVPN. Yeah. I think we might need to change our VPN. Yes. He has a job working. He's doing I think it's genomics. He's working working for a lab. He got a job at a lab in the Davis area doing genomics. Yeah. But I think he's like, I don't know exactly what he's doing. He has to get trained before he can tell us more about what he's doing. Yeah. Yeah, he got another lab job. Exactly. I mean, I was trying to tell him to run for mayor of Davis because apparently that is something he could be very good at. Living in his van, driving around, always knowing what's going on. He's working for the city, cleaning up the streets. I was like, hey, you should run for mayor. You're doing a great job there. Right, Andrew. Yeah, he's got a good lab job, which is, that's fantastic. It is great. Yes. So we are happy for him. And this is not a wah us at all. It's let's just figure out what we need to do to make it keep going. Or oh my gosh. Oh yeah. No, that's what he's doing. No, Justin's not off starting. Maybe he's starting a cell culture in a lab. Cell culture. Oh my God. Waka, waka, waka. That's funny. All right. I am going to head out, go get some sleep. And I hope all of you get some great sleep. Take care of yourselves. Don't bed rot. Try and do things, even if it's just chop wood, carry water, right? Yeah, science island. I do dream of that sometimes now. It was prescient. And let's go take care of the people around us. Thank you, Andrew. Yeah, we're stuck. We're still rocking this platform. We're going to try and do what we can for as long as we can. Thanks for your support. And we Yeah, maybe next time, I don't know, maybe eventually we will have Science Island. Island as in just some land somewhere or on an island in the middle of water. Who knows? I do love that idea. So dream of that. Dream of Science Island. Let's imagine that in our dreams. Sustainability and wonderful learning and people talking science ideas and having fun and being nice to each other. Well, let's be nice to each other. Can we do that? Yep. All right. Be well. Stay healthy. Get some sleep. Stay curious and stay lucky. Have a good night. I'll see you next. I won't see you next week. Good night.